Welcome to today's lecture on neural plasticity and its importance in learning and memory. Neural plasticity refers to the brain's ability to change and reorganize in response to experiences. We will explore synaptic plasticity, molecular basis, forms of plasticity, role of glial cells, epigenetic regulation, neuromodulation, and implications in neurodegenerative diseases. Let's begin. In this video, we will discuss neural plasticity and its role in learning and memory. We will explore cortical remapping, which allows the brain to adapt to environmental changes. Factors such as age, genetics, and stimulation influence neural plasticity, making it important for medical professionals to understand. By understanding neural plasticity, we can develop strategies to treat neurological disorders and promote brain health. In summary, synaptic plasticity is the ability of synaptic connections to change in strength and is essential for learning and memory. Long-term potentiation, LTP, strengthens connections, while long-term depression, limited, weakens them. These processes involve changes in neurotransmitter release, receptor activation, and intracellular signaling pathways. Activation of specific receptors and the involvement of proteins and enzymes contribute to the cellular mechanisms of synaptic plasticity. Long-term potentiation, LTP, is a persistent strengthening of synaptic connections, crucial for learning and memory, through increased neurotransmitter receptors and changes in dendritic spines. Conversely, long-term depression, limited, weakens synapses, helping maintain a balance between excitatory and inhibitory inputs and eliminating unnecessary connections for specific behaviors and memories. Neural plasticity, essential for learning and memory, involves complex molecular processes. Protein kinases including CAMKI and pKa phosphorylate proteins, influencing their function. Transcription factors like CREB contribute to gene expression changes necessary for long-lasting synaptic plasticity. Molecules like BDNF facilitate neuronal survival, growth, and synaptic connection formation. In addition to changes in synaptic strength, neural plasticity also involves structural changes in the brain. Structural plasticity refers to the remodeling of neuronal circuits through the formation and elimination of dendritic spines, the growth of axons and dendrites, and the generation of new neurons in specific brain regions. These structural changes occur in response to learning, exposure to enriched environments, and even after brain injuries. They allow for the rewiring of neural circuits and the establishment of new connections, which are essential for adaptive behaviors and the recovery of normal brain function. Memory consolidation is the process by which newly acquired information is stabilized and stored as long-term memories. It involves a series of cellular and molecular events that take place in specific brain regions, such as the hippocampus and cortex. During memory consolidation, synaptic plasticity plays a critical role. The initial encoding of information during learning is followed by a time-dependent process of consolidation, during which the memory becomes more stable and resistant to interference. This consolidation process is associated with the strengthening of synaptic connections and the reorganization of neural circuits. Hebbian plasticity, named after the psychologist Donald Hebb, is a rule that states cells that fire together wire together. This principle suggests that synchronized firing of neurons leads to synaptic strengthening, promoting the formation of functional neuronal networks. Non-Hebbian plasticity, on the other hand, refers to changes in synaptic strength that are not dependent on synchronous neuronal activity. It can occur through various mechanisms, including heterosynaptic plasticity, spike timing-dependent plasticity, STDP, and metaplasticity, which modulates the threshold for synaptic plasticity. Conventionally, neural plasticity was thought to be most prevalent during early developmental stages. However, it is now well established that the adult brain also retains a remarkable degree of plasticity. This phenomenon, known as adult neuroplasticity, allows for ongoing modifications of neural circuits throughout life. Adult neuroplasticity is evident in various contexts, including learning and memory, recovery from brain injuries, and response to environmental changes. It involves both structural and functional changes in the brain, such as the formation of new connections, remodeling of existing circuits, and the generation of new neurons in certain brain regions. Neuroplasticity has important implications for neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, and Huntington's disease. These diseases are characterized by progressive loss of neurons and synaptic connections, leading to cognitive decline and motor impairments. Understanding the mechanisms of neuroplasticity in neurodegenerative diseases is crucial for the development of therapeutic strategies aimed at preserving or restoring brain function. Research in this area focuses on identifying ways to enhance neuroplasticity, promote neuronal survival, and slow down the progression of these debilitating conditions. Recent research has shown that glial cells, specifically astrocytes and microglia, play a vital role in synaptic plasticity. Astrocytes regulate synaptic connectivity, neurotransmitter levels, and synapse formation and elimination. Microglia, as immune cells, monitor the brain for pathogens and damage, affecting synapse strength and structure. Epigenetic mechanisms play a crucial role in regulating gene expression and shaping neural plasticity. Epigenetic modifications, such as DNA methylation, histone modifications, and non-coding RNA molecules, can influence the accessibility and activity of genes involved in synaptic plasticity. These modifications can be dynamically regulated in response to environmental factors and experiences, allowing for long-lasting changes in neural circuits. Understanding the epigenetic regulation of neural plasticity has significant implications for both normal brain function and neurological disorders. Neuromodulation refers to the regulation of neuronal activity and synaptic plasticity by various signaling molecules and neurotransmitters. By modulating the strength and efficacy of synaptic connections, neuromodulators, such as dopamine, serotonin, and acetylcholine, can influence learning, memory, and other cognitive processes. Furthermore, neuromodulators can dynamically regulate the balance between LTP and limited, promote or inhibit structural plasticity, and modulate the function of glial cells. Understanding the role of neuromodulation in neuroplasticity is crucial for unraveling the complex mechanisms underlying learning and memory processes. Neuroplasticity, the brain's ability to change and adapt, is essential for learning. It involves synaptic and structural modifications, regulated by cellular processes. Glial cells, epigenetic regulation, and neuromodulation also affect neural plasticity, offering potential implications for treating neurodegenerative disorders.